Hello and welcome to today's very first review interview. It's my absolute pleasure to introduce the joint lead authors of this publication, André Görgens from the Karolinska Institute in Sweden and Bas van Balkom from the University Medical Center Utrecht in the Netherlands. We start the interview by finding out a little bit about the author's journey through science so far, what led them to this publication, a bit about the publication itself, and then what it means for the field. So we really hope you enjoy this interview, and please remember to like or comment at the end to let us know how we did.
Yeah, so I entered the EV field quite some years ago when I was working together with Willem Stoorvogel, one of the, I think, pioneers in the EV field. And it was then I was doing proteomic studies, which also is in the end the basis of this paper. And then EVs to me were the unique opportunity to really have an isolated organelle and do proteomic studies on really an isolated organelle. Really, yeah, unique in nature, I think. And then I changed labs, started in the regenerative medicine field, and then, of course, EVs were appearing as a very promising therapeutic agent, and pursued my career really on that, focusing on kidney regeneration, endothelial regeneration, and yeah, finally focusing on the MSCs, which I think are the most promising entities in regenerative therapeutic applications of EVs. Very brief, I think, but that's also complete.
Yeah, great. Thanks. Okay, André.
Yeah, for me, it started during my PhD when I did it in Bernd Giebel's lab in Essen in Germany. He was also a co-author on this paper. And we studied hematopoietic stem cells and how they divide and give rise to all the different blood cells. And we basically did cell biology on those cells and came across some proteins that were distributed asymmetrically and appeared to correlate with sulfate determination of these cells. And these turned out to be tetraspanins to some part. And then we cloned them and saw actually that if you overexpress fusion proteins, that they are like small vesicles also released by the cells. And then that was kind of 2010 or something. And at this point, we didn't have many options to analyze EVs. So I did a lot of flow cytometry before, so I kind of tried to use the instruments I had as well to study EVs or to analyze and quantify EVs, and succeeded with some instruments, especially like this imaging flow cytometry approach. Later on, I then decided, because I became so fascinated by this topic and I really wanted to understand those EVs more, how they facilitate communication between cells. So I went to Samir Lanalus's lab in Stockholm to learn more about EVs and EV engineering, and since then used this multiplex speed-based approach and many other engineering approaches for therapy, and also looking into biomarker discovery. I mean, everybody who knows Bernd Giebel knows that he, like, already since probably 20 years almost, yeah, at least 2007, 8, he started to tell everybody that exosomes or extracellular vesicles are pretty cool and we should have a closer look. And he was driving that forward a lot. So I was heavily influenced by his fascination of those small vesicles, and also introduced to the field by him. So without him, I would probably not have ended up, at least not as fast in this field. So it was very important.
Yeah, you're so right that the people around you are super important in also developing your career. And I think, yeah, similar to André, we entered the field of extracellular vesicles, and they were at that time still called exosomes, in a very, very early stage. And all these pioneers, it was such a nice and friendly community, very, very stimulating. And it was a mix of I could say hardcore cell biologists, also people with a different perspective, really focusing on the therapeutic applications already in the early stages. So we were talking about this community, and everybody was really sharing data and sharing enthusiasm, and yeah, looking at these MSCs. So the mesenchymal stem cells, mesenchymal stromal cells, they can be derived from many different sources, many different culture methods, also many different isolation methods for the vesicle. So it seemed like we were all doing the same, but still we were all doing something different. Still, we did see similar effects, similar characteristics, and that for me was a reason to look into these vesicles. Do they have something in common despite the heterogeneity in MSC source, culturing, EV isolation procedures, etc. And then there was a very stimulating meeting that we had in Singapore, really focusing on the MSCs, and from that we decided to do a proteomics study without touching any pipette. We looked at papers that published proteomics data, and then we saw that despite all the heterogeneity, there was a proteomic signature of these vesicles, and that was really a hint towards, hey, there is really something that we are doing the same. This was picked up by Stefan Wild from Miltony, asking us for input on how can we develop a kit or a tool to robustly assess the quality of EV preparations from MSCs. And I was, of course, very flattered, but also thought, okay, what's in it for us, very bluntly. And their goal to build this robust platform aligned with our wishes to do this inter-laboratory assessment of the kit, taking into account the heterogeneity of these MSCs and the vesicle preparations. And that's when we gathered a nice group across the world to start this effort. And I think, André, you took the lead in this.
I think we both took the lead, kind of. I mean, Bas is definitely more the expert on the MSCs than I am. But when I heard about this, I was happy to join in, and it's really. I always like new toys and new technical methods that we can play with and make it as good as it gets. And the challenge of this was, of course, that we did not sit together and everybody agreed on a very, very specific way how to prepare the EVs, how to culture the cells, how to purify, how to dose the assay, and how to exactly run the assay, because there are different parameters that you can change. But then actually everybody did it like they thought it would be best. And I think this is also one of the strengths of the paper, that there are so many different parameters between all the labs. But at the end, it's possible with this assay to put the data together and normalize it appropriately, and then see the differences and similarities, and identify this common MSC EV signature. So I think that was, it's maybe not like the most fanciest science, but I felt it's like an important thing to do, and I was really happy to see that the results that came out were comparable, even though we didn't align everything like you would do for a big ring trial. And I think that was a good first step towards defining some MSC quality control criteria, or identity criteria.
This interlaboratory effort is something that it's so important, and it's also quite scary. because everybody has his own way of working in the lab and has his own procedures. They often, I'm for myself, I'm very conservative. So I stick to the ultracentrifugation and bone marrow MSCs, and I'm confident that with that approach, I can have nice data and do nice experiments. And it's very scary or maybe frightening to share your data or to compare your data with different labs, because you're always afraid that what you do is only working in your lab. It's something that when you write a systematic review, I think reading systematic reviews, one of the recommendations that you will always see is that there has to be an inter-laboratory effort, because that's hardly ever done. And that's something that's, I think the most important thing we did in this paper.
Yeah, I fully agree. And I mean, I think crucial also has been that we have a narrow question. I've participated in other interlaboratory studies before, and I just feel, of course, it's sometimes more useful if they get bigger and look at every aspect, but it also slows down things dramatically, and it ultimately sometimes doesn't get published or isn't shared with the community. So I think it was crucial for the success of this that we had like a narrow question and a very relatively simple approach. And I mean, in terms of what we found out in the paper, without going into the absolute details, is that we we qualified a method that can be used to robustly assess a certain set of surface markers on MSC-EVs. We found some markers that were very consistent between all MSCs and some that were variable, which is interesting in itself. And we also ran EVs for control from other sources like endothelial and epithelial cells, cell-derived EVs. And you also saw that some of the consistent markers that are on MSC EVs are also to some extent on EVs from other cell sources. So there's not like a black-white pattern that it's only on MSC EVs for most markers. But that's also kind of making sense. And there are common EV markers and somewhat specific EV markers. What we definitely show is that the combination of different ones, so the signature overall, is quite well describing an MSC EV. And a challenge in that study was, for instance, that we also included not only MSC EVs that were prepared differently and different assay protocols, but also MSCs that were cultured with and without platelet lysate, which in itself contain EVs, which then changes the surface signature that we measure on the supernatant. So that was or on the EV preparation. That was definitely one challenge, but even despite this pretty big difference between these samples, we could identify some consensus MSC EV markers or a signature of those. So I think that was kind of the summary. One important aspect also was that the markers we identified, of course, are the ones that were in the panel. So there might be others that are not in that panel. And also it has a certain bias towards higher abundant markers. Because if a marker would be pretty low abundant, then it would be harder to pick it up robustly in all samples, even if you run the assay a bit different. So the MSC EV marker signature is definitely consisting of like the more robust markers because they are more abundant on those MSC EVs.
I think it's very stimulating that everybody who is still using their own approach can pursue on their route. It's clear that doing something different is not doing something wrong. Compared to traveling to an ISEF meeting, some people go by train, some people go by plane. You can even choose to bike. In the end, we all arrive at the ISEF meeting and have a nice time. So the way to get there the way to prepare your EVs can be different, but then the goal and the result is the same. So we're doing things different, but still we're doing things the same.
Yes, I fully agree. And I mean, there are so many open questions still, right? I mean, what we looked at now is like a bulk-based method for MSC EVs. Of course, there are other EVs and there are other methods. Our lab is not an MSC only lab. We work sometimes with MSCs and their EVs, but I mean, there are so many questions. As an example, we are looking into EVs derived from a lot of different cell types and try to find how different are they actually and how similar might some be. Of course, ultimately to identify phenotypic and hopefully functional differences between subsets of EVs that we can then use as biomarkers or exploit for therapies. So I think there's a lot of heterogeneity in the field overall that we don't see yet, just because we don't have the tools or the markers, or took the wrong way to go to the aim, or took a longer way, like we took the bike for some instances, and some things we already know. So it's fascinating, and there's a lot to learn. And I mean, our lab uses a lot of engineering of preparing very specific EVs to deliver a protein or do gene editing, which has helped me a lot also to develop tools. So for instance, what is missing in the field on that side is reference controls that are reliable and calibration material that is really suitable to develop methods. But this is also something that's improved over the last years a lot, and I see a lot of things that we need to do, and I'm excited about the future. I mean, I see for gene correction or for delivering a protein to one specific tissue type, that's something that probably engineering will be much faster and much easier with to treat really very specific diseases. And that's something we're working on in different projects. But on the other hand, unmodified, I mean, just then as an example, we don't know the exact mode of action, how MSC EVs benefit in a certain disease model or disease, right? So there's still a lot to understand. And ultimately, it's very likely to be a multimodal thing where it's not one protein that triggers a signaling cascade or one nucleic acid that is delivered to some cell and then has a function, if that's happening at all. So I think it's probably some kind of complex thing that involves immune stimulation or immune modulation in different ways. And we might never even figure out how it exactly works. But if it works, it's still worth to use, right? I mean, similarly, I think of it like we do, of course, work on gene therapy on different ends. If you just think about cells or CAR T cells for very specific conditions, but we still do blood transfusions, right? So there's no need to engineer everything. Some things just work on its own.
We think we understand. We know that these vesicles have an effect, but still it's a black box. If you look at specific components, you will see that they contribute or maybe not contribute, and that goes for RNAs, for proteins. There are efforts going on in specific targeting those, although we also know that targeting may not be a big issue if you use the natural EVs, because it apparently diseased organs or tissues are more sticky for the EVs. But yeah, also depending if you want to deliver a gene therapy product or a specific protein to a specific cell type or tissue, then engineering may come in handy to keep the vesicles in circulation for a longer time. I think that's one of the challenges to allow them to encounter their targets.
I think it's useful to use those panels all for the biofluids as they are, and of course add more markers in the future to learn even more. We have some unpublished study where we used these kind of panels to look at biofluid EVs, and it works pretty well. And we see interesting patterns and interesting signatures there as well. So the method we described there is not only useful for MSC EVs, I would say. It's for all kind of samples, also other cell types. So there's a lot of markers to uncover that are specific for cell types or enriched in biofluids.
Doesn't necessarily have to be off the shelf. I think you can also combine your own panels and for your specific question, and if you share that, and then also in line with what we did, you can build some confidence in your own preparation, and also make it more comparable with preparation somebody is studying well in the other side of the world. I think the basis, as I said, is this proteomics study that we did. So more like a systematic review like approach, and that's something I worked in a proteomics lab for a few years, in which I also collaborated with Willem Stoorvogel and encountered exosomes at that time, and actually entered the field. But then, that's really some basic research and a big effort. I mean, it's undoable to make proteomics comparisons for every preparation, and I think there so the link is indeed translating these proteomics findings to something that can be used as a quality control, like André is doing in his lab a lot. And that was, and it also comes back to what we said in the beginning. It's such a nice community, and we knew each other for a long time, and we always looking for nice stuff to do together. And this was it. So it was a super nice match. I mean, we like each other. We're friends. We have a beer at the conferences, and but also chat about science. And it's just a nice match.
Couldn't say it better. But I think we also met in Lyon and talked about this the first time at the ISEF meeting, if I remember correctly.
This project, you mean?
Yes.
Yeah. Yeah, that's when we met Stefan from Milton,
Yes.
and he came up with the question whether we would assess this kit, and then as a scientist, you also want to take something away from that. We are not a contract research organization, so we also want to add something to the field and have a nice project together. So we're both happy. I chatted with Stefan yesterday, and he was also really happy with what came out of this study, and they're actually also launching this product in October. Again, it's the super nice and super stimulating community. Saik Yang and Bernd are obviously experts in the field of EVs, MSC EVs, therapeutic EVs. They've involved. André has worked with Bernd. We also previously collaborated with both Saik Yang and Bernd on looking at these therapeutic MSC EVs. So part of the collaboration already exists, without being apparent in publications. We chat, we discuss, and we make plans. We think of things, we make concepts, and then it just came together with this effort.
Yeah, it basically works almost naturally.
Yeah.
And I think Simon was just at the time we were doing these measurements in our lab. He was with a MOVE fellowship from the European organization of local EV societies. He was just having an internship here and had some MSc EVs, so we just measured those as well. So that's sometimes it's just coincidence. But yeah, generally everybody knows each other and works nicely together, and it's a nice community. I guess in brief, biomarker discovery, because it will just come first in terms of diagnostic relevant markers that will be very specific for different diseases and hopefully pick them up early in many cases. I'm not unexcited about therapeutic approaches, but I think this will take a little bit longer to be really widespread used.
Yeah, I'm super enthusiastic about the therapeutic approaches, and but also, yeah, realistically, it's going to be very expensive, I guess, to make these EVs, especially from cultured cells, as like a standard or maybe off-the-shelf therapeutic product. There are many other approaches like the platelet derived vesicles, also other biofluids. We see milk, we see amniotic fluid, where they derive vesicles from, and they show really in small trials positive effects. So there's a lot of effort in going into developing such therapeutic products and also being tested in humans. It's exciting times. I mean, there are trials going on right now, and in Austria, they're very good at these MSC EVs. If we go to the US, then there's many fresh companies with different approaches already doing experiments in humans with different applications, and also successful. So it's going on, and before it's widespread, that will take, now let's say 10 years.